Protein synthesis is the process by which biological cells form proteins. Protein synthesis begins with DNA. DNA, otherwise known as deoxyribonucleic acid, resides in the nucleus of the cell. DNA is composed of small units called nucleotides. Each nucleotide has a phosphate group, a sugar, and a nitrogen-containing base. There are 4 different nitrogen bases:
thymine, guanine, adenine, and cytosine. Thymine always pairs with adenine, and guanine always pairs with cytosine. The bases are also divided into purines and pyrimidines. Purines, adenine and guanine, have 2 carbon-nitrogen rings, whereas pyrimidines, thymine and cytosine, have only single carbon-nitrogen rings. Pyrimidines appear shorter than the purines.
The first process in protein synthesis is replication, where DNA is copied or replicated and 2 complete and identical sets of genetic information are produced.
First, DNA unzips itself with the help of the enzyme helicase. After this, DNA polymerase, another enzyme, bonds free-floating nucleotides to the 2 separated strands of DNA. The same process occurs for the second strand of the original DNA molecule. Finally, 2 identical molecules of DNA result.
The next step is transcription. Transcription is the process by which a DNA message is converted into an intermediate molecule called RNA. RNA, ribonucleic acid, is similar to DNA in structure but has only a single backbone, has the base uracil instead of thymine, and has a sugar ribose instead of deoxyribose.
Transcription begins similarly to replication, where DNA unzips itself and separates into 2 strands throughout the help of the enzyme helicase. 1 strand of the original DNA is used as a template for RNA polymerase to create a complementary strand of mRNA. Lastly, the mRNA, or messenger RNA, strand hang freely and the 2 strands of DNA zip back together. This leads to the last step of protein synthesis, which is translation. This final step occurs in the ribosomes, which the mRNA strand enters after leaving the nucleus. During this process, the individual mRNA codons are translated to amino acids. In the mRNA strand, there are groups of 3 letters that code for amino acids.
These codons attract tRNA, transfer RNA, anticodons with amino acids that are attached.
The tRNA anticodon bonds with the mRNA codon.
Then, the next codon in the mRNA strand attracts another anticodon bearing a new amino acid.
The first tRNA ribosome breaks off from the first codon, and the amino acid it was carrying attaches to the new amino acid. This process repeats itself until a stop codon is reached. Then, the chain of amino acids is released as a protein.